welcome back, everyone. As the chair, as you know, I'm going to keep my remarks short. I'll ask some questions at the end if, in fact, the audience doesn't provide any. It looks like Dr. Om, you're going to present in the oncology area, and that's my background as well. So I'm looking forward to your remarks. So let me pass the screen over to you and look forward to your presentation.
Thank you. Okay, sounds good. So thank you very much, Dr. Rosenberg, for the intro. As a brief intro about myself, I'm Om Sharma. I started my career with research and teaching endocrinology in different medical schools for about 14 years. Subsequently, about 28 years ago, I entered the pharmaceutical R&D world, particularly clinical drug development area. And since then, I have been in the same clinical development area and led successful development of a number of drugs in different therapeutic areas. For example, a couple of hormonal contraceptives, Cyclasa, Merced, they came in market and already off patent a long time ago. Prilogy in urology, sexual medicine, this is a SSRI, it's for premature ejaculation. Dorivax in the infectious disease. And Invokana is a SGLT2 inhibitor for diabetes, and Erleada in prostate cancer. And also a number of NSAIDs and GI products in the OTC area. Also, I had a few successful compounds developed there. And most recently, I have been working in the area of cancer. And in cancer, particularly antibody drug conjugates. Of course, I have worked on the hormonal contraceptives for prostate cancer, which is Erleada, was approved a couple of years ago. Then currently I'm working on antibody drug conjugates. And these are for multiple indications for breast cancer, for prostate, for colorectal, lung, etc. And so therefore I thought I will speak about the ADC compounds, their development in the cancer treatment. Mm-hmm. And that brings to mind this topic. And I need to know how to...
Yeah, if you hide that, you see on the bottom right, there's a little banner at the bottom where it says you stop sharing and hide. If you click on the hide to the right, right there. There you go. Now you can advance your slides.
Oh, okay. Thank you.
Sure.
So I have divided my, I think I will have about 20 minutes or so to speak and then leave some time for question and answers. So I have divided my you can call it a talk or a discussion. Basically, I want to have it as a interactive discussion. In these parts, I will introduce the compound and the ADC and what are the key elements of ADCs and ADC technology, and then how does ADC work for the cancer treatment, and then identify a few challenges what we still have, and there are a lot of other challenges might be there, and what we think as a future direction of antibody drug conjugates development. So with that, ADC concept appears to be very simple. So this is a kind of 4 parts. 3 of them is the complex, it's comprising of a monoclonal antibody. It's conjugated to a biologically active cytotoxic payload, and those could be small molecules, and via a chemical linker. This chemical linker is this is basically biodegradable, either it cleaves or it degrades by itself in the tumor environment. And it is directed towards these are 3 parts, and the 4th part is directed towards the antigen expressed on the cancer cell surface. And I will talk about why these are important components, even the concept being very simple. But this is this complex molecule that require very careful attention to each and every component of these, all 4 components. And 5th one is the drug-antibody ratio. So I will go on the selection of appropriate target, which is antigen, is on the cancer cell surface, monoclonal antibody, and cytotoxic payloads. So as of now, those could be divided into two classes. One of them are microtubule disruptors. Another are the one who does DNA damage, and that is the way to kill the cancer cells. And then the appropriate linker, which is again in two groups, can be classified in two groups: cleavable and non-cleavable, and the antibody drug ratio. This is a kind of schematic depiction of this complex. This is antibody basically, which targets the cell surface on the antigen or the receptor, or antigen as a receptor, which is carrying cytotoxic drug, which as I said, those could be either the microtubule disruptors or DNA destroyers linked by a chemical linker. So this is the whole complex. It attaches to the target cells on cancer cells. So we give a brief history of the development of ADCs. It's a little bit interesting because the original concept of drugs that would target tumor cells and spare healthy tissue was conceived in 1900 by German Nobel laureate Dr. Paul Ehrlich. And then, of course, as usual, it took so many decades, and the first ADC, gemtuzumab ozogamicin, by the name of Mylotarg, was approved in 2000, and this was for the CD33 positive acute myeloid leukemia. But it was unfortunately voluntarily withdrawn in 2010 because of the toxicity issues, and FDA requested to withdraw that. Then the second one was brentuximab vedotin, or Adcetris. This was for Hodgkin's lymphoma, came in 2011 by FDA and EMEA by 2012. So you can see that there is a considerable time when they are the developers could be successfully developing these ADCs. It's a long time taken there, and I'll talk about those, why it is such a complicated compound. Then the third one was adutrastuzumab emtansine, or Kadcyla, in 2013 for HER2 positive breast cancer. Fourth one was inotuzumab ozogamicin, and this was for acute lymphocyte leukemia, approved in 2017 by both EU Commission and FDA. And then came Mylotarg again, reintroduced in the market in the same year, 2017. After addressing the dosing issues, what they did is 9 milligram per kilogram divided into 3 doses, 3 milligram per kilogram, and that's how it took care of toxicity issues. And then subsequently, some new, more recently, drugs approved like polatuzumab vedotin, also known as Polivy, for the large B-cell lymphoma in 2019, and more recently, trastuzumab deruxtecan Nixi, which is known as Enhertu. It's from our company, Daiichi-Sankyo, approved for metastatic breast cancer in 2019, and most recently for the breast cancer, sacituzumab govitecan in 2020. So there, as usual, the development takes a while. So for first or early ADCs, they encounter a lot of major obstacles. Those were mainly, they have low blood residency time, low penetration in the solid tumors, and low payload potency, because they were normally developed from the chemotherapies which were used without any covering. So their toxicity was, the efficacy was not as enough as that should have been with the ADCs. Then they developed the first generation ADCs. In those one, for example, doxorubicin, this is BR96, KS14 methotrexate. In this one, they conjugated the payload with the murine antibodies and via a non-cleavable linker. So these are sort of early on compounds, and the issue with that is, one thing is the antibody is not specific, therefore not humanized. The other thing was these non-cleavable linkers were not, again, as appropriate to release the drug in the microenvironment of tumor cells. So example is gemcitabine, ojogamicin. And what they did is they used a very potent calicamycin derivative and with a humanized antibody. So this reduced the immunogenicity and improved efficacy, but it still had some drawbacks, like linker was not stable enough, and because of high percentage of unconjugated antibody, this did not have the desired efficacy. So poor CMC and high toxicity was still an issue. Then came second generation ADCs, and then again they improved on all these 3 or 4 components, and adotrastuzumab, emtansine, and inotuzumab, ojogamicin. These are example of the second generation, but it still has drawback. It has off-target toxicity, fast clearance, and the competition with unconjugated antibodies. Then came the third generation ADCs, and those are the site-specific conjugation was introduced in this one, and that would provide, or that has provided, a kind of uniform CMC. And these days, ADCs had a homogeneous ADCs with drug to antibody ratio anywhere from 2 to 4, and the most recent are having 8, which I will talk later on the significance of this. And the one I have been working, or Daiichi-Sankyo has been working, like NHER2, and there is also HER3 targeted antibody, and this ADC, which has ratio of 1:8. So these are the, again, cartoon of explaining that components of antibody drug conjugate. So this antibody drug conjugate is conjugated on the cell surface to antigen on the tumor cells, or receptors on the tumor cells. And this has 2 types of conjugation types. So either this is non-specific conjugation, which is through lysine or cysteine residues, or it is site-specific conjugation, which is either through, these are 3 different mechanisms or technologies: engineered cysteine, or enzymatic conjugations, or incorporation of unusual amino acids. And this antibody contains the cytotoxic drugs, which are 2 classes of drugs. Either they are targeting microtubules, and these are the examples of, or mostly used microtubule disruptors, is auristatin derivatives, or metranzinoids, tubulizins, or DNA disruptors like calicamycins or diocarbamycins. These are mostly used payloads, cytotoxic drugs. These are linked through this chemical linker, and this chemical linker is also can be classified into classes, either non-cleavable or cleavable. So cleavable takes the advantage of lysosomal degradation, then everything disintegrates once in the tumor environment, microenvironment, and releases the drug. Or cleavable linkers are either acid sensitive, which take advantage of low acid microenvironment in the tumor cells and releases the drug, or lysosomal protease sensitive, which are degraded by lysosomal degradation or redox, like disulfide bond, and they release the drug in the system. So why these all components are very important are that. So I will just briefly go over that. Antigen selection criteria is very important because it should have high level expression on the tumor cell surface and low level expression on the healthy cells to have a targeted maximum targeted therapy and minimum cytotoxicity in the general cells or healthy cells. So this should be also expressed on the tumor cell surface, especially for the solid tumors. So it has easy access to the tumor cells. And the rate of internalization and route of intracellular trafficking is also very important component of this, because what happens, because this needs this antigen and this complex integration and internalization cells, but also some of them, they can be cell permeable. And because of that permeability, they could also, once they have internalized in a tumor cell, they can escape that cell and can go to another neighboring cells, even without they have antigen on those cells. And that is called bystander effect. So it increases the efficacy. However, it could also have some safety issue. Antibody selection is again like molecular weight has to be appropriate. Normally, it should be around 150 kDa in order to be appropriately bound to the cell and move around in the system. The other component is that specificity should be also optimum, and the affinity is the optimum. Affinity is considered somewhere anywhere 0.1 to 1 nanomole per liter. If it is too tightly bound, then it will have issue of releasing, and if it is too loosely bound, then it will again have the toxicity or CMC PK issue. The payload should be the cytotoxic drugs. It should have high toxicity to tumor cells. One of the reasons the initial ADCs didn't work or did not have efficacy was that they were using the same cytotoxic drugs which they were using naked. And because because of the toxicity, their potency was kept deliberately lower, not to be intolerable. But when you use those in the ADC, they didn't have the desired effect. The reason is that when you inject an ADC in the body, only 2% of that drug reaches to the tumor cells. So therefore, the cytotoxic drug has to be very, very potent in order to have the efficacy. And then they should have also functional growth, or this can be derived to the conjugated with the antibody, and should have stability in the physiological condition. So when it is being injected, it is circulating in the blood or in the circulation system. It should be stable. It should not release there and the payload in the system. Otherwise, it will cause toxicity. But once it goes in the tumor cell, it should be quickly releasing that. That's where comes the linker technology. So this linker should be, again, it should be stable in the extracellular fluid, but it should be pretty fast to release in the cell inside. And as I mentioned earlier, these could be divided into cleavable or non-cleavable linkers. Here are some of the examples I have already mentioned. The drug antibody ratio is again very important, because the number of payload molecules per antibody is called the ratio. So if it is too low, it will have the reduced potency. If it is too high, then it will diminish the stability because of the hypophilic nature of the drugs, and then it will reduce the stability, and therefore it has to be optimum ratio. Here are some examples recently approved drugs. I won't go in those. There are at least 8 to 10 drugs already in the market. And briefly talking about the mechanism of action, about the ADCs, I have mostly alluded to that, but this is just to explain that systematically what happens, that this ADC compound, which is antibody linked with linker with payload, and then it attaches to the either receptor or antigen on the cell surface. What happens after that? It internalizes, goes into the endosome, and then disintegrates in the lysosome using the mechanism what I described earlier. Either they degrades, or there is because of the properties of linker, it will use either pH, or it will use, like for example, in cathepsin increases in the tumor cells. So if this is enzymatic degradation, then it will be using cathepsin, will be disintegrating. Then the cytotoxic drug is released in the cell, and then based on the mechanism of that particular cytotoxic drug, either it does DNA damage or it works through the microtubule disruption. And here is what happened. Some of the free drug, based on the cell permeability, depending on the property of this drug, whether it is cell permeable, it will migrate to the neighboring cells, and irrespective of the antigen on these cells or receptor on these cells will go and kill them. So that is also called bystander effect, and this improves efficacy of certain compounds. So having said that, it appears mechanism appears very easy, but we have a lot of challenges as of now. So the toxicity issue, most of these drugs still have the neutropenia, peripheral neuropathy, some of them, and thrombocytopenia, GI, and some of them have ocular toxicity and hepatic dysfunction. These are the common side effects, major or concerning side effects. And then some of the drugs, they have grade 5 interstitial lung disease. And this is not a high percentage, but still it needs to be brought down. It is somewhere in like 10% or so. And some drugs, as I mentioned, have high grade ocular or neurological toxicities. We still don't have a very good handle on biomarker assessment, biomarkers for patient selection, because it's important to have that. And yes, there is a lot of development going on in this one to have optimal selection of patients. So developing these biomarkers, and as of now, often they lack validated assays to define antigen positivity. So for example, we use like IHC, which is relatively I wouldn't say very vague, but it is still not very precise. And so we need to develop that further. And there is still a lot of room for developing specific antibodies. And some of the preclinical models are not very predictive of clinical effect. So actually, I have I think maybe a couple of minutes left. Quickly, we'll go over the future directions or possible things which could be done. Is that there is new potential targets. They need to be worked on. So they are other than tumor-associated antigens. Examples are given here. Smarter vehicles for payloads need to be developed. ADC prodrugs circulating in an inactive form to be activated after proteolytic cleavage by tumor microenvironment proteases, which is up to certain extent we are doing, but still needs some improvement. Alternative delivery systems could be developed, and there are new classes of antibodies like paratopic or bispecific antibodies. So you can have two different drugs sent to the tumor, and that could take care of the resistance mechanism, because one you can target, another you can take over the resistance mechanism. Payload alternatives, the different payload needs to be developed, and combination strategies are the best thing as of now, which could be meaningful. I will skip that. And in summary, as I mentioned, that these ADCs are empowered antibodies. Basically, they are designed to harness the targeting ability of monoclonal antibodies by linking them to potent cytotoxic payloads via chemical linker, and these exert their activity by selecting binding of the antibody tumor, internalization, and lysosomal degradation and release of cytotoxic payload, leading to tumor cell death. While concept of ADC is relatively straightforward, the design and synthesis takes really very hard work and very intellectually developed titrating of different all 4-5 components of ADCs to make it efficient ADC. And then ideal ADC should have all these components: monoclonal antibody, a cytotoxic drug, linker, and appropriate DAR, very wisely titrated. With that, thank you very much. I took a little bit more minutes than I thought. So
no, you're actually fine. We have another 5 minutes or so to go for your presentation. I haven't seen any questions come through, so maybe we'll just have a little chat while we wrap the session up. And so, as you think about this delivery system, to me, the tumor microenvironment constantly changes. And so, as you think about the ways to circumvent through that environment, What do you do with the prospects of your treatments? Is that why we don't see as many, because you can't solve for that? Or have you figured out a way to navigate through the microenvironment to get the payload and to get the target pretty well hit now?
So there are still like continuous improvement is going on in that direction. So what happens? Yes, as you said, microenvironment changes. I would say, based on some of our work, that there are I would say a lot of challenges. So for example, when any chemotherapies, after a while there is a mechanism of developing resistance. And one thing is, since, as you said, microenvironment changes all the time in the tumor microenvironment, so how can you overcome of that resistance mechanism? So as of now, I mean, one of the things in the ADC development area people have been doing is combining. And actually that goes same for the chemotherapy environment also, to combining different different compounds or chemotherapies. So for example, we are targeting in HER3 targeted tumors. And once this HER3 biology is HER2 is relatively well known, but HER3 is not as much known as HER2. So what happens after a while, you stop seeing the efficacy. What happens? So if you use another cytotoxic drug, that could either resynthesize or somehow it enhances the HER3 synthesis, either on the cell surface, or it is recycling that HER3 receptor from it comes on the surface. But it is still unknown how does it do, but it takes care of that resistance, and it will give you another whatever duration to have the same drug or compound functioning again.
All right, and I guess I can keep asking a few questions until we get to the end of our time period. So what about, like, let's just say, hiding it to some degree to avoid the macrophage detection?